Hello everyone, welcome to the next episode of Micro Talks. This episode is based on some practical aspects of microbiology. In this episode, we will be studying the direct microscopic count, which is a rapid method to count the bacterial cells in both stained as well as unstained preparations. Let us now look at what is meant by a direct microscopic count, or DMC as it is also known. DMC is a quick method that enables us to count the microflora in a given sample. It is used to quantify microorganisms in a sample by observing them and counting them directly under the microscope. It is often used as a platform test in the dairy microbiology. There are two types of methods that could be used for a direct microscopic count. One of them is the breeds count, which is a method that uses stained preparations. Normally they are stained by crystal violet, or you could even use stains which can differentiate between live and dead cells. The other method of carrying out a DMC is the use of a hemocytometer or a Petroff-Hausser's chamber, which can be used in unstained preparations. The hemocytometer is also known as the Neubauer chamber. Let us look at the construction of the hemocytometer. So the hemocytometer was invented by Louis Charles Malasses, and it is a glass slide, specialized glass slide with a precisely etched pattern on its surface. There is a specialized cover slip which is thicker than our normal cover slip. So this etched pattern allows the counting of cells within a known volume of fluid. A rectangular indentation is etched into the center of the slide here, and there is this groove here which is 0.1 millimeters in depth. So it creates a chamber with a precise depth. This chamber floor is engraved with a grid of perpendicular lines, as you can see in this figure. This perpendicular square, this square is further divided by parallel lines and perpendicular lines, and there are at the four corners the WBC squares, and in the center there is the RBC square, which is used for counting. This hemocytometer is normally loaded with a precise volume of the sample using a pasture pipette or a micropipette with sterile tips, and the sample is loaded in the groove of the hemocytometer slide. So this is basically the construction of the hemocytometer slide, how it is constructed. Let us now look at the requirements for this particular experiment. So you need a Neubauer chamber or a hemocytometer slide with a cover slip, a pasture pipette or a micropipette with sterile tips, and the sample that has to be counted. The sample may be appropriately diluted. However, at the time of the final calculations, the dilution factor should be taken into account. The hemocytometer slide can be cleaned with a brush and covered with a cover slip. If we are using the WBC chamber, then like I said before, we have to count all the 4 WBC squares, each being further divided into 16 squares. If we are using the RBC square, then we have to count the corner 4 RBC squares and the one in the center. So you take a clean hemocytometer slide, cover it with a cover slip, and using a micropipette, place a small drop of the suspension in the groove such that it can spread on the slide. Then you have to place the slide in the petri dish containing a moist blotting paper and allow the slide to settle. So this is basically you are keeping it in the petri dish so as to avoid the sample from getting dried. Thereafter, mount the hemocytometer slide under the microscope and you count the cells using the high power lens. So if we are using the WBC chamber for counting the cells, then the calculation is like this. So the volume of 1 WBC square is 1 millimeters in width, 1 millimeters in length and 0.1 millimeters in depth. So the volume of 1 WBC square is 0.1 millimeter cube. Therefore, the volume of 4 WBC squares. Why 4? Because we are counting 4 WBC squares. So the volume of 4 WBC squares therefore is 0.4 millimeter cube. Now, the total number of cells in the 4 WBC squares is equal to M. Let me tell you at this point that we are not considering average number of cells. We are taking the total number of cells. So W1, W2, W3, W4, which is the total number of cells, which is equal to M. So 0.4 millimeter cube has got M cells. So therefore 1000 millimeter cube or 1 ml will have 2500 times M cells by a simple calculation. So the total number of cells that you get in the 4 WBC squares is multiplied by 2500. To this final number, if there is a dilution factor, then you have to multiply the dilution factor. This is the calculation for the WBC square. Now coming to the calculation for the RBC square. Volume of 1 RBC square is equal to 0.2 millimeters by 0.2 millimeters in width and length and 0.1 in depth. So this is equal to 0.004 millimeter cube. So the volume of 5 RBC squares therefore becomes 5 times 0.004, which is equal to 0.02 millimeter cube. Now here we are counting 5 RBC squares. So that is the 4 corner RBC squares and the one in the center. So therefore the number of cells, again here we are considering the total number of cells. So the total number of cells in the 5 RBC squares R1, R2, R3, R4 and R5 is equal to N. Now 0.2 millimeter cube contains N cells. So 1000 millimeter cube or 1 ml will contain 50,000 into N. So whatever is your total number of cells in the 5 RBC squares is multiplied by 50,000. And this final count is then later on multiplied by the dilution factor if the sample has been appropriately diluted. Let us look at the applications of the hemocytometer. Obviously, because there are WBC and RBC squares, it is used to count the WBC and RBC from the blood. It is used as a cell, count the cells in the cell culture. It is used to count the number of sperms in a given semen sample. And in the brewing industry, it is used for the preparation of yeast at the time of preparing the inoculum of the yeast that has to be added to the fermentation in the beer brewing industry. Let us now look at some of the advantages of using a hemocytometer. It is a quick and rapid method of counting the cells. The disadvantages are that it gives you a total count. It cannot distinguish between the live and the dead cells. Every cluster of cells is counted as one cell, and if the cell lies at an intersection between two squares, then it is counted as only one. So these are some of the drawbacks of using the hemocytometer. However, the fact that it is very rapid, it's a rapid method, overcomes these drawbacks. It's a simple method. There is no great technical know-how required for this. And you simply have to mount the hemocytometer under the high power and start your counting. So that's all for now in this episode of Micro Talks. Please do like, share and subscribe to my channel of Micro Talks. Looking forward to bringing you more episodes. Thank you all for watching this episode.